transamination process and deamination process. So in the transamination process, we talked about the transferring of amino group from an amino acid to a alpha keto acid, which will generate one more thing. Okay, that's called transamination. So here we discussed about the enzyme also. Those two enzymes are SGOT and SGPT. What is SGOT stands for? Serum glutamate oxalate transaminase. What is SGPT stands for? Serum glutamate pyruvate transaminase. So these two are having the other names also. What are those? SGOT is also called as AST. AST, that means aspartate transaminase. Okay. And SGPT is also called as AL, that's called alanine transaminase. So these two enzymes are mainly involved in the transamination process, with following by the enzyme by the B complex vitamin. So that is called vitamin B6. So vitamin B6 is also called as pyridoxal phosphate, the active form of the vitamin B6. Okay. So all these coenzymes are the active forms of the B complex vitamins only. What are the coenzymes we are talking? All these are the active forms of the B complex vitamins. So like the thymine, the active form is thymine pyrophosphate, riboflavin, FMN, FAD. So these things are acting as a coenzymes. Okay. So here we also discussed about the SGOT, where the also called as aspartate transaminase. Here this is the amino acid aspartate, which is donating its NH2 group to an alpha ketoprotein called the alpha ketoprotein, so to form the one more structure called the oxaloacetate and the glutamate. So it means the glutamate is the product which is formed up here in the transamination process. So at the same time, by the help of the enzyme B6 and all, so at the help of them we also discussed about the aspartate transaminase and alanine transaminase also. So in the alanine transaminase, again the alanine is donating its amino group to an alpha ketoprotein and forming a glutamate, and the alanine is converting into the pyruvate. Okay. So alanine is converting into the pyruvate and alpha ketoprotein is converting into the glutamate. So that is the product which is formed up here. That means glutamate is taking up the amino group. Okay. So this glutamate will undergo deamination process. What is deamination process? Deamination process means removal of amino group is called deamination. So here we also discussed about about these two enzymes, where it is present and what is the role of these enzymes. Okay. So coming to the SGOT, it is called as serum glutamate oxalate transaminase, which is mainly present in the heart and also present in the liver. Okay. So whenever any cardiac dysfunction is observed, at that time the SGOT levels will also elevated. Okay. So the normal range is 55 to 40 international units, and the same thing there also nearly 40 international units. SGPT also the same thing, 5 to 40 international units per liter. So these two levels will be elevated in the liver cirrhosis or liver diseases. Okay. So and we discussed about the deamination process also. What is deamination? Removal of amino group is called deamination. So the glutamate which is formed up here in the transamination process, the glutamate which is formed up in the transamination process have to undergo deamination process. So by removing the amino group. So whenever the glutamate is undergo deamination process by the help of the enzyme called glutamate dehydrogenase, but in between one intermediate form will be observed, that is called alpha aminoglutarate. So this alpha aminoglutarate is converting into alpha ketoglutarate. Okay, and the ammonia is released. So like this, that after the transamination and deamination process, the NH3 group is released. So which have to enter into the urea cycle, because this ammonia which is becoming more and more, that leads to problems that called encephalopathy, blurring of speech, blurring of vision, slurring of speech will be observed there. Whenever the ammonia levels are there, these ammonia levels it's totally toxic to our body and toxic to our brain mainly. Toxic to the body at the same time, toxic to the brain mainly. So which leads to blurring of vision, slurring of speech, at last coma, and finally death will be observed to that persons. Okay. So we also discussed about the different types of animals like uricotelic, ureotelic, and aminotelic. What is uricotelic? So the amino group is released in the form of uric acid. That is called uricotelic animals. Example, the reptiles are the examples for the uricotelic. What is aminotelic? Aminotelic means, ammoniotelic means it will dispose the ammonia directly into the water, like the terrestrial animals or the mainly the fishes, aquatic animals. All these things will come under the ammonotelic. Okay, last one is called the ureotelic. So this ureotelic means it have to convert into urea, because ammonia is totally toxic. It have to convert into the non-toxic form called the urea. So we people, that means the primates and the man, will come under the ureotelic. Okay. So up to now in the last class we discussed here. So coming to the main thing, today's topic, urea cycle. What is urea cycle? As I already told you, because the ammonia is totally toxic to our body, so it have to convert into urea. So for that one cycle is required. That means that is called the urea cycle, which is taking place in the liver only. It's taking place in the liver only. Okay. The urea is the end product of protein metabolism. After protein metabolism, what is the product form? The ammonia. So ammonia will convert into the urea. So that's why what the end product? The urea is the end product of protein metabolism. Okay. So you can tell what is about uric acid. Uric acid ఎక్కడ ఫామ్ అవుతుంది? The protein metabolism కాకుండా purine metabolism. అంటే purines ఉంటాయి కదా, AMP, GMP, CMP. So these purines, that is the nucleotides. So then when they are degraded, it will convert into uric acid. But proteins will convert into urea. Okay, so the protein is the urea is the end product of protein metabolism. So the nitrogen amino acids which are converted to ammonia, which is totally toxic to our body. So at last the ammonia is converting into the urea. Here the urea is a nitrogen containing substance. That means NPN, nitrogen, non-protein nitrogen substances. Okay. So the urea is one of the non-protein nitrogen substances, because at the same time in the practical lab also we will have the practical examination, practical labs also will be there. Okay, ఎప్పుడైనా practical వచ్చారా? So they will start the practicals. Okay, so the practicals ఏం చేస్తామంటే in the urine sample also we have to check the urea levels. We will check the urea levels in the urine sample. Along with the urea, they will also check the uric acid levels also. Not only in the urine sample, it is also present in the blood sample also. Okay. అయితే ఇక్కడ చూడండి please, first of all listen. Whenever the kidney failure is observed, at that time the most sensitive index for kidney failure is the creatinine. Most sensitive index for kidney failure is creatinine. అయితే along with this creatinine, the urea levels will also elevate. Along with the creatinine, the urea levels will also elevate. So the creatinine ratio, urea normal range అంటే 20 to 40. The normal range of urea is, please listen, I will show you all things. Urea normal range is 20 to 40 milligram per deciliter. Along with the urea, we also prefer to test that creatinine levels. Okay. So whenever the person who is having any kidney failure, okay, but dialysis ఇంకా వెళ్ళలేదు, సో వాటి creatinine levels first of all check చేస్తాం. Creatinine levels ఎంత ఉంటాయంటే up to 0.8 to 1.2 వరకు normal range. 0.8 to 1.2 is the normal range. So these normal ranges would be elevated up to 14, 16, 18 వరకు వెళ్ళిపోతాయి. అంటే కిడ్నీ ఫెయిల్ అయ్యింది అంటే ఫస్ట్ ఆఫ్ ఆల్ ఏం ఎక్కువ ఇంక్రీజ్ అవుతుందంటే creatinine levels. Along with the creatinine, the urea levels will also elevate. సో ఇప్పుడు మీకు శాంపుల్ వచ్చిందనుకో, బ్లడ్ శాంపుల్ వచ్చిందనుకో, సేరం శాంపుల్, వాడి కిడ్నీ ఫెయిల్యూర్ ఉంది, ఆ సేరం శాంపుల్ చాలా తక్కువ వచ్చింది. సో ఫస్ట్ టెస్ట్ వీ హ్యావ్ టు పెర్ఫార్మ్ ఈస్ ద క్రియాటినిన్. ఓకే, క్రియాటినిన్ చెక్ చేసిన తర్వాత అప్పుడు యూరియా లెవెల్స్ చెక్ చేస్తాం. ఓకే, సో యూరియా లెవెల్స్ ఎందుకు చెక్ చేస్తామంటే క్రియాటినిన్ కండిషన్ ఎలాగో పెరిగింది, దట్ ఇస్ అలాంగ్ విత్ దట్ ద యూరియా లెవెల్స్ విల్ ఆల్సో ఎలివేట్ ఇన్ ద కిడ్నీ ఫెయిల్యూర్ పర్సన్స్. సో నాట్ ఓన్లీ దట్, మామూలుగా కిడ్నీ ఫెయిల్యూర్ కాకుండా అదర్ కండిషన్స్లో కూడా యూరియా లెవెల్స్ విల్ ఆల్సో ఎలివేట్. ఓకే, క్రియాటిన్ అనేది సెన్సిటివ్ ఇండెక్స్. ఓకే, దాంతోపాటు యూరియా లెవెల్స్ విల్ ఆల్సో ఎలివేట్. యూరియా లెవెల్స్ విల్ ఆల్సో ఎలివేట్. సో ఇఫ్ పర్సన్ ఇస్ నాట్ హ్యావింగ్ ఎనీ కిడ్నీ ఫెయిల్యూర్, అంటే కిడ్నీ ఫెయిల్యూర్ ఏం లేదు, నార్మల్ పర్సన్ ఇస్ ద నార్మల్ పర్సన్, బట్ హీస్ హ్యావింగ్ సమ్ బర్న్స్, సమ్ other conditions like diarrhea, vomitings and conditions, or any renal stones present in the sample. So at that time, the urea levels will also elevate. అర్థమైందా? సో క్రియాటిన్ అనేది కిడ్నీ ఫెయిల్యూర్లో పెరుగుతుంది. యూరియా లెవెల్స్ అదర్ దాన్ ద కిడ్నీ ఫెయిల్యూర్, సమ్ అదర్ కండిషన్స్ ఆల్సో ద యూరియా లెవెల్స్ విల్ ఆల్సో ఎలివేట్. సో లైక్ వామిటింగ్, డయేరియా, ఆ కండిషన్స్లో పెరుగుతాయి. ఎనీ స్టోన్స్ కానీ ఉన్నప్పుడు కూడా అట్ ద టైమ్ ఆల్సో ద యూరియా లెవెల్స్ విల్ ఆల్సో ఎలివేట్. So in all these three conditions, in the kidney failures, in the any vomiting diarrhea conditions or any skin conditions, at the same time any renal stones are observed, at that time also the urea levels will elevate. Okay. So the urea is a non-nitrogen content substances. It is synthesized in the liver, transported to kidney and excreted in the urine. So as I already told you, the ammonia is totally toxic to our body. So this ammonia have to convert into urea. So where it have to convert? It will by the help of the urea cycle. Where it is taking place? It is taking place in the liver. It is taking place in the liver. So that ammonia will convert into the urea. So this urea will be entering into the kidney, from where it will excrete out. Okay. Just write this point only. Few points. First, urea is the end product of protein metabolism. So the ammonia is converting into urea, and urea is an NPN. NPN means non-protein nitrogenous substances. And come to the last point, it is synthesized in the liver, and then it is transported to the kidneys and will be excreted in the urine sample. NPN means non-protein nitrogenous substances. Okay. So ఈ ప్రాక్టికల్స్ అని చేస్తారు. ఇన్ ద non-protein nitrogenous substances, you will get a urine sample. With the urine sample, we have to perform the test belong to the urea. That means the urine sample containing the urea or not. Right. So the structure of urea is NH2 C double bond O NH2. Or structure of urea, the structure of urea is NH2 C double bond O NH2. So the first NH2 is arriving from the ammonia. That means what the ammonia which is precipitate. So this ammonia gives the NH2 group, and C double bond O is obtained from carbon dioxide, and the remaining third NH2 group will be obtained from the aspartate. The aspartate will donate one more NH2 group here. So, have you heard the word Krebs cycle? What is Krebs cycle? Krebs cycle is also called as tricarboxylic acid cycle, also called as TCA cycle. That means when of the carbohydrate, that means we are taking polysaccharides or disaccharides or monosaccharides, whatever the food you are taking up. So that have to be converted into the glucose molecules. So the glucose will convert into the pyruvate by the help of glycolysis. Again, this pyruvate will enter into the TCA cycle. How it will enter? Directly it won't enter. The pyruvate have to convert into acetyl-CoA. The pyruvate will convert into acetyl-CoA. That acetyl-CoA will enter into the TCA cycle, also called as tricarboxylic acid cycle. So the tricarboxylic acid cycle is also called as Krebs cycle. Okay. What is urea cycle? Here one more person is added there. That's called Hanslet. Krebs-Hanslet cycle. So but please don't be confused between the TCA cycle, that means Krebs cycle, and Krebs-Hanslet cycle. Krebs-Hanslet cycle means it's a urea cycle. Okay. Krebs cycle means the TCA cycle. Okay. So it is also called as one more cycle called the ornithine cycle, and it is having a 5-step process present in the mitochondria. Some enzymes, so that in the liver itself only, because it is a major tissue. Okay, so the liver is having the mitochondria and cytosol. In the liver, liver lo మొత్తం liver in the liver cells lo mitochondria, cytosol lo mitochondria lo 2 enzymes. Okay, 2 enzymes are present in the mitochondria and remaining 3 enzymes present in the cytosol. So the urea cycle is a 5-step process. Urea cycle is a 5-step process. In that 2 steps will be by the help of the mitochondria, because their enzymes are present only in the mitochondria. After that, it will enter into the cytosol. In the cytosol, remaining enzymes are there. They will activate and convert into the urea. Asana? See. So this is urea cycle. So as I told you, the urea cycle is taking place in the liver. In the liver also the mitochondria and cytosol. What are the 2 steps which are involved? The first 2 steps are involved in the mitochondria and the remaining steps are in the cytosol region. Okay. So here you can see the ammonia which is liberated out. Ammonia ekkada vachindi? Transformation, deamination process అయిన తర్వాత ammonia is liberated there. So that ammonia will bind with the carbon dioxide molecule, okay, by the help of the carbon dioxide molecule to form a structure called carbamoyl phosphate. For this, one enzyme is required that is called carbamoyl phosphate synthase 1. Okay, here it requires 2 ATP molecules. The 2 ATP molecules are converting into 2 ADP plus PI. And what is this NOT? And what is NOT? Okay, N-acetyl glutamate. N-acetyl glutamate is the coenzyme activator here. N-acetyl glutamate is the activator. Okay, so this carbamoyl phosphate, as I already told you, the urea cycle is also called as ornithine cycle. Urea cycle is also called as ornithine cycle. So this ornithine will bind with the carbamoyl phosphate. The ornithine will bind with the carbamoyl phosphate by the help of enzyme called ornithine transcarbamylase to form a structure called citrulline. Up to here, it is totally in the mitochondria only. That means these 2 enzymes. What are those 2 enzymes? Carbamoyl phosphate synthase 1 and one more enzyme called ornithine transcarbamylase. These 2 enzymes are present in the mitochondria only. Okay. So first of all, what the ammonia we are getting it from? Ammonia is getting from the deamination process. After trans-deamination process, the ammonia is liberated. So to this ammonia, the carbon dioxide molecule is binding up here to form a structure called carbamoyl phosphate. So for this carbamoyl phosphate formation, it requires the ATP. How many ATPs are required there? 2 ATP molecules are required. So this 2 ATP are converting into ADP and PI. At the same time, what the enzyme required here? The carbamoyl phosphate synthase 1. Okay. And what NAD stands for? N-acetyl glutamate. So N-acetyl glutamate is the activator here. So our activator untene our carbamoyl phosphate synthase 1 act avutundi, so that it convert the carbon dioxide and ammonia to form the carbamoyl phosphate there. Okay, so this carbamoyl phosphate will bind with an ornithine which is coming from the cytosol region, and it will bind with the by the help of the enzyme called ornithine transcarbamylase. It forms a structure called citrulline. Okay, up to citrulline. So all these reactions, the first two reactions are taking place in the mitochondria only. So this citrulline can enter into the cytosol. The citrulline which is formed up here can enter into the cytosol. Is the citrulline is converting, is forming up there by the help of the enzyme ornithine transcarbamylase. So this citrulline is converting into the arginosuccinate. What's converting is converting into arginosuccinate. So for this the enzyme is required, is called arginosuccinate synthetase. మనం ఫస్ట్ ఒక చెప్పాం NH2 C double bond O NH2 అని స్ట్రక్చర్ చెప్పాం. అవునా? ఒక NH2 ఎక్కడ వస్తుంది చెప్పాము? From the ammonia, and carbon dioxide వస్తుంది carbon dioxide is giving the C double bond O. ఇంకో అమోనియా ఎక్కడ వస్తుంది చెప్పాము? Aspartate. So that is the aspartate here. So the aspartate is added to the citrulline molecule in the cytoplasm region by the help of the enzyme arginosuccinate synthase to form structure called arginosuccinate. Okay, here also it requires the ATP molecule which is converting into AMP and PPI. Okay, so this arginosuccinate by the help of enzyme called arginosuccinase releasing the fumarate group. What is releasing? Releasing the fumarate group. This fumarate group will enter into the TCA cycle. A fumarate activity again it is entered into the TCA cycle. So that's why there is a link between the urea cycle and TCA cycle. Urea cycle ki TCA cycle is an interrelationship is there, so that the arginosuccinate by releasing the fumarate group, this fumarate group is entering into the TCA cycle to form a structure called arginine. So that means the arginosuccinate by the help of enzyme arginosuccinase, it will remove the fumarate group out and form structure called arginine. Okay, so this arginine by the help of enzyme called arginase, it will release the urea molecule out. Okay, here what we require, the urea is required, the urea is evolved out. Okay, so whenever the urea is evolved out, the arginine is converting into the ornithine. Again the same cycle will takes place. Okay, so one more time. So normally what's happening there, the ammonia by the help of transamination process and by the help of deamination process, it is the ammonia is liberated out. So this ammonia have to enter into the liver so that it will convert into the urea. Okay, so in the liver also we are having two different types of places where that is called the mitochondria and second is the cytosol. In the mitochondria we are having two enzymes, so that is responsible for the formation of the citrulline. Okay, and remaining these three enzymes are present in the cytosol region, also called cytoplasm. Okay, so in the cytosol region. So what's happening there? The ammonia is binding with a carbon dioxide molecule to form a structure called carbonyl phosphate. For this, the two molecules of ATP is required, which is converting into AMP and PPI. Okay, so for that one enzyme is required. What is that enzyme? That's called CPS1. And take a chapter. CPS1, carbonyl phosphate synthetase 1 and chapter. Okay, so that means one more enzyme is also there called CPS2. CPS1 is there, CPS2 is there. So CPS2 is involved in the nucleotide metabolism. CPS2 అనేది nucleotide metabolism లో మనకి కనబడతా ఉంటుంది. Okay, so that's why we are calling it as CPS1. So with the help of an activator, what is the activator there? N-ester glutamate is the activator. So that we convert into a carbonyl phosphate. This carbonyl phosphate have to bind with an ornithine. Okay, so that's why we will call this as an ornithine cycle also. So this ornithine will bind with a carbonyl phosphate by the help of enzyme ornithine transcarbamylase to form a structure called citrulline. Okay, so this up to here is all the reactions are taking place in the mitochondria only. So whenever the citrulline is forming up there, the citrulline only can enter into the mitochondria, from the mitochondria to the cytoplasm. The citrulline is the only structure which can enter into the cytoplasm. Okay. So when a citrulline is entering the cytoplasm, the citrulline will bind with a one more amino acid, so which is providing the NH2 group to the urea. Second NH2 group to the urea. Ever aspartate is giving it. So the aspartate is giving this NH2 group to the citrulline by the help of the enzyme arginosuccinate synthetase to form a structure called arginosuccinate. So this arginosuccinate, by the help of the enzyme arginosuccinase, it will release the fumarate group, which will enter into the TCA cycle. Okay, so that's why the urea cycle and TCA cycle both are called as a bicycle. Okay, Adi cycle, Idi cycle. So both will call it as bicycle. Okay. So when a fumarate is released, it will convert into the arginine by the help of the enzyme. One more enzyme called the arginase present in the cytosol. It will release the urea out. So at last, what the product we are forming up there? The urea is the end product, which is formed in the protein metabolism. Okay. So when it's converting, the arginine is converting back into the ornithine, and the cycle will go on like that. That's what we call it as urea cycle. Right. Textbooks, you are having the reactions and all. So our structures ఏమీ అవసరం లేదు. So just simply write these things, that will be enough for you. Even for the MBBS also, structures are not at all required. Don't buy the structures and don't be tense with that. discussing about the same cycle only. All these slides are discussing about the same slide only. That's called urea cycle. So what are the role of it and so what I told is called the bicycle, as I already told you. So this is called the urea cycle, where the ammonia and carbon dioxide combine to form carbon, phosphate, and discipline and all. So here, where the aspartate is obtained from, that means the auxiliary state in the TCA cycle will convert into aspartate. So this aspartate is donating its NH3 group to the cyclin to form the arginosuccinate. At the same time, after this arginosuccinate, we are releasing the fumarate group. This fumarate is entering into the TCA cycle. So that is the interrelation between the urea cycle and the TCA cycle. And one more thing I forgot to talk about this. So that means here NH4 to C double bond O dash NH4 NH2. So that means one NH3 from the ammonia and C double bond from the carbon dioxide. Where this carbon dioxide is obtaining from? The carbon dioxide is obtaining from the same TCA cycle only. So whenever the respiration taking place, or the metabolism taking place here, so at that time the carbon dioxide molecule is evolved out from the TCA cycle called the tricarboxylic acid cycle. That carbon dioxide will bind with the ammonia group here. Let's see here. The carbon dioxide molecule is binding to the ammonia group. So like that, all these three, NH2, which is coming from the ammonia, C double bond O from the carbon dioxide, which is from the TCA cycle, and third ammonia from the aspartate. That aspartate is obtained from the oxaloacetate. Okay, the TCA cycle. Practice. Disorders of urea cycle. Main function of urea cycle is to remove the toxic ammonia from the blood as urea. Okay. Okay, so here I am talking about hyperammonia. What is hyperammonemia? What is hyperammonemia? Means whenever the urea cycle is not forming up, so due to defect in the enzymes, I told you different types of enzymes are involved in translation of the ammonia to the urea. Okay, so whenever any of the enzymes are in defect, any of the enzymes not function properly, at that time the ammonia will be like that only. Ammonia cannot convert into the urea. Any defect in any of the enzyme, okay, mainly the first enzyme is there. So if it is deficient there, so at that time the ammonia cannot convert into urea. So that is called hyperammonemia. Okay, this is called hyperammonemia. So defect in the metabolism of conversion of ammonia to urea, that is urea cycle, leads to hyperammonemia or ammonia intoxication. So when this condition will be observed, mainly due to the inherited disorders of the urea cycle enzymes. Any inherited disorders in the enzymes, that means enzymes are not functioning properly, are not synthesized properly, at that time this hyperammonemia will be observed. Along with that, so as I told you, the ammonia urea cycle, what is the tissue there? Where it's taking place? Taking place in the liver. If liver is not functioning properly, any liver cirrhosis observed, at that time the ammonia loss will also elevate. So here that's why acquired disorders like liver diseases, severe renal diseases. Why renal diseases? Because the ammonia have to convert to urea. At that time, urea have to enter into the kidney. Okay. So if this kidney is not functioning properly, at the time even the ammonia will also elevated up. Okay. So because more amount of will be observed. That's called acquired hyperammonemia. So these are different metabolic defects in the urea cycle. So I have told you any of the enzyme, first enzyme which is called hyperammonium type 2, that means carbonyl phosphate synthase CPS1 అంటున్నాం కదా, that's called carbonyl phosphate synthase 1. So if this enzyme is deficiency there, that condition is called hyperammonemia type 1. If second enzyme which is called the ornithine transcarbamylase enzyme is deficient, then we will call it as hyperammonemia type 2. And here with the third enzyme which is called arginosuccinate synthase. Okay, so this enzyme is absent, then we will call the citrullinemia. That means the citrulline will be becoming more and more because citrulline have to convert to arginine. So here see, the citrulline have to convert into arginosuccinate. So if this enzyme is absent, that leads to citrullinemia. Okay, so at the same time, arginosuccinase. What's arginosuccinase? Arginosuccinase is here. Okay, that means arginosuccinate have to convert into arginine. For the enzyme required is the arginosuccinase. If this enzyme is absent, so then it leads to arginosuccinate urea. Okay, so at the same time, ఇక్కడ రాసేసుకోండి, అక్కడ రాసుకోండి, మీ ఫిగర్ వేసిన దగ్గర, that will be better. సో ఇలాగ మీ రికార్డ్స్లో కానీ మీ టెక్స్ట్ బుక్స్లో కానీ టోటల్గా ప్రతి పాయింట్ ఇవ్వరు. ఓకే, దట్ మీన్స్ ఇఫ్ దిస్ ఎంజైమ్ ఇస్ డెఫిషియన్సీ డైరెక్ట్గా వాళ్ళు ఇందాక చెప్పినట్టుగా స్లైడ్ వేస్తారు. ఓకే, సో కానీ ఎక్కడ మనకి బ్లాక్ అవుతుంది, దట్ మీన్స్ ద carbonyl phosphate synthase 1 is absent, that condition leads to hyperammonemia type 1. If this enzyme ornithine transcarbamylase is enzyme deficient, that leads to the hyperammonemia type 2. And if the citrulline arginosuccinate synthase is absent, so then it leads to citrullinemia. If the enzyme arginosuccinase is absent, then more amount of arginosuccinate is forming up there, which is called arginosuccinate urea. If this arginine have to convert to arginine, further the enzyme required is the arginase. If this enzyme is deficient, more amount of arginine will be forming up there. It's called hyperarginemia. Okay, that's what I have written here, same thing. Write these symptoms. If increased levels of ammonia will result in the slurring of speech, blurring of vision, convulsions, any vomiting, nausea, neurological defects, and mental retardation, at last the coma and death will be observed in the person who is having hyperammonemia. That means increased levels of ammonia will be observed here, which leads to these conditions called the slurring of speech, blurring of vision, convulsions, nausea, vomiting, and neurological deficits, because the ammonia will enter into the brain. Ammonia can easily enter into the blood-brain barrier. అందుకని బ్రెయిన్ ఎన్సెఫలోపతి, that will lead to coma and death. ఎప్పుడైతే అమోనియా లెవెల్స్ ఎక్కువైపోతున్నాయో అమోనియా యూరియాగా కన్వర్ట్ అవ్వాలి. యూరియా కన్వర్ట్ అవ్వట్లేదంటే అమోనియా లెవెల్స్ ఎక్కువైపోతే ఏమవుతుందంటే ఈ అమోనియా బ్రెయిన్లోకి వెళ్ళిపోతుంది. దట్ విల్ ఎంటర్ ఇన్ టు ద బ్రెయిన్. సో దట్స్ వై వీ విల్ అబ్జర్వ్ ఆల్ దీస్ త్రీ కండిషన్స్ లైక్ స్లరింగ్ ఆఫ్ స్పీచ్, బ్లరింగ్ ఆఫ్ విజన్, okay, and convulsions, nausea, all these things will be observed here. At the same time, the coma and death will be observed due to the high amount of ammonia present in the sample. write this point only for the treatment. The first point for the treatment, we will have to give the intravenous administration of sodium benzoate and phenyl acetate. So these two are administered here for the as a treatment. 5.1 try సరిపోతుంది. So these sodium benzoate and phenyl acetate will condense with the glycine and glutamate to form a water soluble product, so that the ammonia can be converted into the water soluble product and can be easily excreted out. So that's why what the doctor will do, they will give the sodium benzoate and the phenyl acetate. So these two will bind with the ammonia molecule and forms it into an easily excretable molecule structure. Okay, so that the ammonia will be excreted out. Right, the normal range in the is 10 to 40 milligram per deciliter. Right, the third point, used for evaluating the kidney. Used to screen the evaluating the kidney. అంటే చెప్పాను కదా, ఫస్ట్ ఆఫ్ ఆల్ మనం కిడ్నీ ఫెయిల్యూర్ పేషెంట్స్కి ఎక్కువగా యూరిన్ లెవెల్స్ చెక్ చేస్తూ ఉంటాం. అలాంగ్ విత్ దట్ we also check the creatinine levels. So first of all we will discuss about the we will do the creatinine, and after that we will perform the urea. Okay, so both the creatinine levels and the urea levels will be elevated in any of the kidney failures. First third point and it's all important. So in your examination also, or the practical examination also, we have to check the urea levels in the given serum sample. First of all, what they will do in the practical lab, they will give the serum sample. So they will give the blood sample. With the blood sample, we have to check the take the serum. How we can check the serum sample from blood sample? ఏం చేస్తాం blood sample తీసుకున్నాక ఒక పేషెంట్ వచ్చాడు, కిడ్నీ ఫెయిల్యూర్ పేషెంట్ వచ్చాడు. సో వాళ్ళ దగ్గర మనం బ్లడ్ శాంపుల్ కలెక్ట్ చేయాలి. హౌ వి కలెక్ట్ బ్లడ్ శాంపుల్? ఎలా కలెక్ట్ చేస్తాం? బ్లడ్ శాంపుల్ కలెక్ట్ చేస్తుంది తెలుసా? బ్లడ్ శాంపుల్ కలెక్ట్ చేసిన తర్వాత దాన్ని సీరం ఎలా సెపరేట్ చేస్తారు? బై సెంట్రిఫికేషన్ ప్రాసెస్. సో ఫస్ట్ ఆఫ్ ఆల్ బ్లడ్ శాంపుల్ తీసుకున్న తర్వాత ఇట్ షుడ్ బి క్లాటెడ్. క్లాట్ అయిపోయిన తర్వాత, దట్ మీన్స్ వీ హ్యావ్ టు కీప్ ఇట్ ఫర్ ప్రెజర్ ఫర్ 20 minutes. Nearly 20 to 30 minutes we have to keep it up, so that it will get clotted. After clotting it, we have to keep it in the centrifuge machine and we have to centrifuge it. After centrifugation, all the dense particles will get down and the main serum will be on the upper side. Okay, so from that serum we have to perform the test called the urea test. So the normal range of urea, the normal range of urea in serum sample is 10 to 40 milligram per deciliter. So as I already told you, the urea is one of the indicator for kidney failure. So the urea levels will also elevate in other conditions also. What are those conditions? Those conditions called the pre-renal conditions, renal conditions and the post-renal conditions. What are pre-renal conditions? అంటే కిడ్నీకి పైన మనం ఏం చెప్తాము, కిడ్నీ ఫెయిల్యూర్ అనుకుంటున్నాం. కిడ్నీ ఫెయిల్యూర్ అంటే కిడ్నీకి పైన ఏంటిది? ప్రీ-renal. Okay, next renal, after post-renal. So pre-renal conditions ఏంటంటే, so any dehydration like diarrhea, vomiting or any burns, shock, okay, massive hemorrhage. First minimum 2 to 3 points రాయండి. Pre-renal లో just write the first 2 points. Dehydration like diarrhea, vomiting, and second point burns and shock. సరిపోతుంది. Keep it heading as pre-renal. At the time of examination, practical examination also, they will ask this viva. So what are the pre-renal conditions, what are the renal conditions and what are the post-renal conditions. Okay, so pre-renal లో first 2 points రాయండి, renal లో మొత్తం రాయండి, minimum 4 points రాయండి. Acute glomerulonephritis, chronic glomerulonephritis, nephrosis in any polycystic kidney. All please write all the 4 points in the renal conditions. And come the last one called the post-renal conditions. The post-renal conditions means any obstruction in the urinary blood flow. The urinary flow. So at that time we can see the creatinine urea levels will be elevated. Keep a star mark upon this. So they may ask you in your examination also. Even in the practicals also you will be having this one. Any of the kidney failures, it may be a chronic glomerulonephritis, it may be acute glomerulonephritis, any polycystic kidney or any nephrosis. All these conditions, the urea levels will elevate. Okay. In the pre-renal condition, what are the pre-renal conditions as I told you? Any burns or any shocks అంటే కిడ్నీ ఫెయిల్యూర్ లేకపోయినా సరే యూరియా లెవెల్స్ ఎలివేట్ అవుతాయి. కిడ్నీ ఫెయిల్యూర్స్ ఉన్నా యూరియా లెవెల్స్ ఎలివేట్ అవుతాయి. కిడ్నీ ఫెయిల్యూర్స్ లేకపోయినా యూరియా లెవెల్స్ ఎలివేట్ అవుతాయి. ఆ కిడ్నీ ఫెయిల్యూర్ లేకుండా ఉన్న కండిషన్స్ ఏంటవి? దట్ మే బి ప్రీ-renal conditions or any post-renal conditions. Pre-renal conditions ఏం చెప్పాము? Any diarrhea, vomiting, shocks, burns, all this will come under the pre-renal conditions. What are the renal conditions? Any chronic glomerulonephritis, acute glomerulonephritis, polycystic kidney, okay, in any of nephrotic conditions, all these conditions, the creatinine, urea levels will be elevated. And come to the third point. What is the third point? Post-renal conditions. And the kidney that is tarvata. Okay. So what are those? Any obstructions in the flow of the urine. Urine by exit out from any obstruction, any stones, renal stones, the urea levels will elevate there. Okay, so that's what we have written here. Obstruction to the flow of urine and prostate enlargement. When the prostate is enlarged, at that time also the urine will not be excreted out. Okay, so that's why the hyperuremia will be observed here. All stones in the urinary tract, tumors in the bladder. All these conditions in that your urea levels will be elevated. What is the normal range? Normal range is 10 to 40. మీకు కొంచెం ఈజీగా ఉందనుకుంటే 20 to 40 అయినా పర్వాలేదు. బేసింగ్ అపాన్ ద కిట్ they will tell you. So 15 to 40 అంటారు, 20 to 40 అంటారు, 10 to 40 అంటారు. So please keep in mind 20 to 40 milligram per deciliter. Okay. So just past one. Here, if people are here, but if you are going to the out of countries, మీరు కానీ ఇక్కడ చదవకుండా బయటికి వెళ్ళి ప్రాక్టీస్ చేసేటప్పుడు అంటే అవుట్ ఆఫ్ కంట్రీస్ ప్రాక్టీస్ చేసేటప్పుడు దే వోంట్ టాక్ ఇన్ ద యూరియా అని పేరు చెప్పరు. దే విల్ కాల్ ఇట్ యాజ్ బన్, బ్లడ్ యూరియా నైట్రోజన్. బ్లడ్ యూరియా నైట్రోజన్ అంటే వాట్ బ్లడ్ యూరియా నైట్రోజన్ మీన్స్ హాఫ్ ఆఫ్ ద యూరియా నైట్రోజన్. హాఫ్ ఆఫ్ ద యూరియా నైట్రోజన్ ఇస్ కాల్డ్ బ్లడ్ యూరియా నైట్రోజన్. అంటే మనకి యూరియా కానీ 40 milligram per deciliter వచ్చిందనుకో, బన్ వాల్యూ ఎంత అవుతుంది? హాఫ్ అవుతుంది, 20. ఓకే, సో మీరు అక్కడికి వెళ్ళేసరికి మన వాల్యూస్ తగ్గి లో వాల్యూస్ వచ్చేస్తున్నాయి కదా అని అనుకోవద్దు. సో దట్ మీన్స్ హాఫ్ ఆఫ్ ద యూరియా నైట్రోజన్ ఇస్ ద వన్ వాల్యూ. దే విల్ కాల్ ఇట్ యాజ్ బ్లడ్ యూరియా నైట్రోజన్. ఓకే, ఈవెన్ మనకి ఇక్కడ క్లినికల్ ల్యాబ్స్లో కూడాను విల్ క్యాల్కులేట్ ద వన్ వాల్యూ, బట్ వీ విల్ మల్టిప్లై ఇంటూ 2.1 అంటే ఇంటూ 2తో మల్టిప్లై చేస్తాం. ఓకే,